So as Pamela mentioned, my work combines social psychology, sociology, particularly social network analysis, and neuroscience, in particular functional brain imaging, fMRI. And what I'll be talking about today are the neural foundations of social relations in everyday, real-world social groups. So one thing that most people, most sociologists, psychologists, neuroscientists can agree on is this idea that human brains and our species more generally evolved to navigate our uniquely complex group dynamics, and that these dynamics, these relationships, are embedded within complex social network structures. But while there have been many advancements in the ability to probe neural processes or brain mechanisms that are involved in all kinds of psychological phenomena, one of the problems is that if you're somebody like me who's interested in studying social relationships, it's very hard to do that in an MRI machine, right? So there's just a technical difficulty of how we study participants, human subjects, in this kind of research. And there's also kind of a theoretical or conceptual framework of studying individuals as independent islands. So we recruit randomly from the population, often in social psychological research or cognitive neuroscience, and then we have a bunch of participants just like this person, but they have no relationship to each other. And that's actually a feature of many of these studies in order to get experimental control over some of these kinds of characteristics. So my approach to this research that I'm doing here in the PSSN program comes at it from a different kind of stance, an interpersonal context that is emphasized in social network analysis. And so as a basis, a kind of basic paradigm that I've been using in this research project is to recruit actual groups of people who know each other, who exist in the real world. And these are social networks, I should clarify, that interact face-to-face, not online. And the reason for that is that there are lots of complex kinds of dynamics that arise in face-to-face groups that we know more about in terms of a long history of social psychology work on small groups and also neuroscience research on animals that don't use online social networks. The other thing that feel free to laugh at corny jokes. There are a few more embedded in here. So the other feature of this kind of research design is that it allows us to measure participants' relationships to each other. And that's not with any kind of sophisticated technique. That's just literally by asking people, how much do you like so-and-so? How close do you feel with them? We also collect all kinds of other measures, but using this data about how people feel about each other and their social connections, we can then quantify their relationships and their position within the social network structure. And then we can see how that evolves over time. And finally, the groups that I've studied, in each case we've used the same round-robin neuroimaging paradigm that incorporates each group member who we're studying, both as a perceiver, so that's somebody who is put in the scanner, provided that they consent to that and don't have metal in their body. And we also incorporate them in the research design as a target of social perception. So we take pictures of all of our participants and then we show those pictures as stimuli during the fMRI scan. So today I'll be talking about two kinds of social relations, liking and reciprocity. So liking, I don't need to define it for you. One of the things that social scientists have been really interested in, I would say not just social scientists, but people generally who have ever been in a kind of group setting, is that sometimes we end up becoming friends with certain people in a group, not others. And to some extent, there are principles in sociology that I'll talk more about in a few slides that help us explain some of these kinds of reasons for why we might end up befriending certain people and not others. But we don't know about the underlying mechanisms. And so brain imaging, or the promise of brain imaging, is that it can help social scientists, social psychologists, sociologists explain individuals' personal liking preferences using methods that have been developed in cognitive neuroscience. And reciprocity, there's a sense in which individuals start off in a group with kind of unique feelings about people. And I'll show you actual data about this from the groups that we've been looking at. But just to foreshadow where we're going with this, I'm interested in how two individuals' idiosyncratic liking sentiments when they first meet each other become mutually reciprocated or coupled at a dyadic level, and how liking goes from being an individual level phenomenon to a dyadic phenomenon. Okay, so the basic question here is trying to predict group members' ultimate patterns of social relations. And so we're trying to see if we can predict who ends up liking each other. And this is a question that has intrigued social psychologists and sociologists alike for the better part of the century. And there have been many different approaches to try to predict interpersonal liking. Here we're using a language of attraction, which is not to be confused with romantic or sexual attraction, but more about the kind of attraction that brings social atoms together into metaphorically bond to create these kind of interpersonal or social bonds. And sociological frameworks, of which I'm going to focus on social network analysis, they tend to emphasize structural forces. So that could be things like homophily. People who are of similar age, race, socioeconomic status tend to like each other. An enemy of an enemy is a friend, that kind of transitive property of social relationships. But in terms of the amount of variance that we can explain as social scientists about who we end up liking, the most salient kind of social force is a really simple one: reciprocity, which is that I like you if you like me, or you like me if I like you. It also goes, of course, for negative kinds of sentiments as well. And there have been theoretical postulations by social psychologists influenced by behaviorism in the 50s and 60s in psychology that put forward theories that interpersonal attraction, again, this is just platonic liking between people, can be conceptualized in terms of reward value. And this kind of construct of reward value changed depending on the theorist, but you can think of this kind of on an experiential level, with the idea that you may have had the experience, you may have been told before that if you smile during an interaction that is otherwise uncomfortable, I'm going to try it right now. So if I smile, that from a lot of social psychological research has been shown that it will tend to make me like you more in this kind of social interaction. So we can think of that as an interpersonal kind of effect, but also it's been shown that when you smile at people, they tend to like you back also. And so that simple act of smiling, because it's an interactive kind of expression, it can affect both how much I like you and how much you like me. Okay. What I'll be talking about today is fMRI work that leverages the fact that we can measure bold activity, oxygenated blood flow that has to do with neural activity in the brain. And I'm going to just be focusing on a couple of brain regions that putatively are involved in reward value. And I'll make some caveats up front that I am not wed to this being brain systems that are explicitly for reward value. We're working backwards here in some ways from a social psychological theory to map that onto some kinds of brain activity. And the true test of whether that was successful in some ways is whether it allows us to predict the outcomes that we care about additionally. So I'll be focusing on two brain regions, the ventromedial prefrontal cortex, or the medial orbital frontal cortex, and ventral striatum. These are brain regions that have been implicated in processing reward value. They are involved in reinforcement learning, motivating affiliative behavior. they're involved in lots of different kinds of things depending on the kind of neuroscience research that you do, whether it's in humans, animal models, and what you study in particular, what you think that these brain regions do. But they are definitely involved in processing the reward value of various kinds of social and non-social stimuli. We know that from both animal, non-human animal and human neuroimaging research. So while I said before that we tend to study in human neuroimaging, we tend to study individuals as isolated kind of islands, in other branches of neuroscientific inquiry, particularly in a lot of the work on neurobiological models of hormones and neuroendocrine responses between mother and infant, between mates in rodent models, partner preference kinds of sexual bonds in, for example, monogamous prairie voles. And so those kinds of dyads have actually been studied in animal models where the researcher will study both the mother and the infant, for example, and see how an input on the mother's side will affect both the mother and the baby. For any of you who have a child or a spouse or any kind of human interaction partners, you know that an input like how much they have been sleeping affects their outcomes, like their satisfaction in the next day, but also yours, right? In the same way, your inputs, how much you sleep, affects how happy you are the next day, and also probably your interaction partners. And so that kind of intuition has been appreciated by a lot of neuroscience researchers working with animal models where they can actually manipulate these things, go into this brain system, whether with kind of hormonal inputs, with oxytocin or vasopressin, or doing dopaminergic kinds of manipulations that we can't do with people. One thing that has been done with people looking at these brain regions in terms of neuroimaging is looking to see what happens when in these brain regions when participants look at non-social consumer goods. So this is a DVD of Madagascar. Participants would look at things like that DVD, a poster, another DVD, a CD, a book. This is a little bit dated based on the kind of stimuli that they're using, but a DVD is basically a movie. And so what happens is that as participants are looking at these kinds of stimuli, researchers are looking to see what's happening in these regions of interest in the brain, and they're finding that based on the neural response in these brain regions to these consumer goods, they can predict better than chance individuals' idiosyncratic kinds of preferences. So we know that these brain regions also respond to stimulus characteristics, like people who symmetrical faces will tend to elicit more activation of these reward-implicated regions, but also there are these kinds of idiosyncratic preferences. So Madagascar, the DVD, appeals to some people more than others, and that will be reflected in brain activity. Okay, so the study that I'll talk about today is kind of a proof of concept study to see if we can leverage these neuroscientific insights to predict how future liking ties will develop in a newly acquainted group. The design of this study, and I'm happy to talk more about this in the questions afterwards about the specific participants that we had in this study, because I do think that there are questions of how this would generalize. We took advantage of a really neat kind of natural experiment that happened when one of the co-authors on this paper had developed a 9-week intensive summer program for college students who were interested in organizing Walmart workers. So these are social justice-minded college students who applied to this program. They were going to be living and working together throughout a summer doing really intensive work, and they amazingly volunteered to also be a part of this brain scan study. So during the first few days after they all came to New York, we scanned them within the first 3 days after the group was conceived, when they all came together at New York. We took fMRI scans to measure these kind of the responses of these brain regions, the vmPFC and ventral striatum, to every group member. and we measured how much each of the participants liked each of the other participants. We did this in a few different ways that all produced similar results. What I'll be talking about today is a measure that went from not at all to very much, that you, on a visual analog scale, rated along that dimension how much you like somebody, and then we converted that to a number from 0 to 100 based on the percentage of how much you filled up that bar. And so we asked that question of how much you like each of the other participants throughout the summer, and at the conclusion of the program, a little more than 2 months later, we again got the same measure of how much they like each of the participants. Then what we're doing with them in the scanner is just a basic face perception task. So we're showing the participants pictures of themselves and each of the other group members for 1 second at a time, and that's interleaved with a randomly jittered fixation cross, and they push a button every time they see a group member, and then about 10% of the time they see this face, which is all the faces put together, and that's just to keep them awake because this is not the most exciting task. And the reason that we have such a short presentation time of these faces is that we want to try to see how these brain systems are responding as close as we can to spontaneously without having a task where they need to evaluate how much they like this person. It's just the face popping up for a second. What is it eliciting? That face goes away, another face comes up. What is it eliciting? And as the schematic shows, we have each of the participants embedded both as a perceiver and as a target. And so this allows us to deal with some confounds that, for example, if this face is more symmetrical than this one, this face more symmetrical than this one, it might elicit more reward system activity from everybody. And so we can partial that out, even though we can't manipulate it because everybody sees everybody else. We can remove some potential confounds that have to do with just differences in face structure or individual differences in how, for example, my striatum might just be very reactive to all people. Okay, so first just to look at what our dependent variable is, to look at liking, because liking is something that develops in different ways in different kinds of group contexts. So I think it's important to first get a sense of what actually happened in terms of liking ties for these participants in the study. So first thing is to show here we have T1 on the left side, T2 on the right side. So again, this is within the first 2 days of them meeting each other. This is 9 weeks later after they've lived and worked together. What this is showing is what is driving most of the variability, the variance in people's liking ratings. So this ego effect has to do with about a quarter of the liking variance has to do with the fact that some people just generally don't like anybody, and some people tend to like everybody a lot. These alter effects, you can think of them as popularity. Some people tend to be liked by everybody in a group. Some people tend not to be liked by anybody, and that also accounts for about a fifth of the variance. You can see that the majority of variance at time 1 is everything else that's left over. So people have unique levels of liking, of gravitational kind of draw between them that they experience between different people. Some of the other work that I've done is looked at this measure of popularity, and that was actually what this study in some ways was initially conceived to do. But the fun thing about studying people in the real world is that their liking patterns are not tailored towards what you necessarily want to study. And in this group, you can see that at time 2, very little of liking is actually structured around consensus effect, popularity, of how much people agreed about a certain group member being liked. And so, if anything, liking is actually getting a lot more idiosyncratic over the course of the summer. And so we can look at these kind of individual level plots. This is showing for each participant at time 1 and time 2, each of the liking ties. So this participant in the beginning liked everybody between 50 and 100, and in general they tended to like most of the other group members more at the end of the summer, except for this one participant that they ended up liking a lot less. We can also look at the same data organized by the recipient of the liking ties, and we can see, for example, that there are some participants, for example, this one here, that in the beginning everybody liked them okay, and they were pretty stable. One person liked them a lot less. So, when you're working with groups this small, you can really start to, in some ways, there's a danger of trying to overfit a model, both conceptually and with the data. And so we can talk at the end about what the implications are of the fact that this model fits some people better than others. Another important thing about what happened with liking in this group between T1 and T2, the x-axis here is showing liking ratings relative to a mean. One thing that might be surprising is that people actually didn't really end up liking each other any more or less from time 1 to time 2. So the average of people's liking ratings at time 1 was about 64% of the scale across. At time 2 it was about 66% of the way across. So 2 points along a 100-point scale over a summer. These people live together, they work together. This is, obviously the story is not just about a mean level of liking that changed. What you can see here is that at time 1, a lot of the liking ratings were heaped around the mean, and that there wasn't a very big distribution of liking ratings around that. Whereas at time 2, a lot of the liking ratings that were heaped around the average here are now appearing at the tails. So liking ratings became more extreme in both directions. We have more people who really don't like each other, more people who really like each other than we had at time 1. And finally, what happened with liking here, which I think we should have expected, but I did not, is that reciprocity, which is one of these kinds of really basic facts about social interaction, about how groups are structured, you can see that it's actually not apparent at time 1. So what we have on the x-axis is ratings of how much one person liked another one, here labeled kind of arbitrarily actor to a partner, and how much that partner likes that actor. So at time 1, if I want to know how much Federica likes me, it doesn't help at all to know how much I like her. Those things are almost entirely unrelated. At time 2, you can see that there's actually a really strong relationship here where the more that you are liked, the more that you like somebody. And the question is, in some ways, because this is what we're trying to predict at time 2, this measure here of how much one person likes another, or how much one person likes another, because that is so reciprocated, what we're trying to predict is now not just an individual level phenomenon, how much do I like this person, that person, that person, how much do I like Madagascar, this CD, that book, but now we're trying to predict these kinds of reciprocal relationships. We're predicting a dyadic outcome. And so what that means is that there is a different kind, the fact that this data is structured in this way is both a problem statistically, but it also produces a nice feature, which is that it allows us to use models that were developed for studying people embedded in relationships. So this is research that social psychologists tend to do to predict something like relationship satisfaction with the examples that I was giving before. Here we'll look at it just with liking. So we'll have 2 hypothetical group members, Anita and Buddy, and what we see here is just the actor effect. So this is an intrapersonal pathway, how much Anita's initial liking of Buddy predicts her future liking of Buddy. And you can see that's a significant effect. That's something that we know from social psych research showing thin slice kinds of effects that there's going to be a correlation across all of you for how you initially perceived me in the first few seconds with how you'll feel about me at the end of this talk. And so social psychologists have focused on the fact that there is stability here, but you could also point out that actually the vast majority of variance in future liking is not explained by initial liking, so there's a lot of change. And we can also then look at the interpersonal pathway, the partner effect of how Anita's initial liking of Buddy predicted Buddy's future liking of Anita, or said differently, how much Anita ends up liking Buddy is also predicted by how much Buddy initially liked her. And so this kind of actor-partner interdependence model shows us how actor's liking of partner, which initially was not related to partner's liking of actor, how that at the end actually becomes reciprocated because an input, for example, if we could somehow manipulate Anita to like Buddy more, that would end up predicting that she will like Buddy more and also that Buddy will like her more. And so that will bring their liking outcomes closer together. And so this actor-partner interdependence models explains how individual inputs end up becoming coupled at a dyadic level. And so we also did that with brain activity. And this is a little bit weird because this is not something that is typically done with brain imaging data, but what we looked at is vmPFC and ventral striatal activity. We got the same effects for both, so I'm lumping them together here to show how Anita's neural response to the brain activation pattern that she got when looking at Buddy predicted how much she would end up liking Buddy, but it also predicted how much Buddy would end up liking her. And these effects are approximately equal, both the intrapersonal and interpersonal pathways. So we could also flip that around and say, if we want to try to predict who will ultimately like me, we can do that based on people's neural responses to me initially, just as well as with my neural responses to you. And these effects also hold when we control for both partners' initial liking as well as both of their neural variables. So to sum up the results that we've found so far, this kind of interpersonally elicited activation of these reward-related brain regions predict, it's a loaded term, so I'll say it foreshadows. We use it to predict. It doesn't mean that that's what the brain regions are doing, but we as researchers use that brain activity to predict who will ultimately like us, or who we will ultimately like, and who will ultimately like us. And in terms of reciprocity, it also helps us understand something that sociologists haven't really been interested in, the mechanism that produces reciprocity, which one of the suggestions implied by our results are that dyad members, so any two members of a group, their initial conditions, both their explicit liking ratings and their implicit brain measures that we used, they interdependently shape, or at least interdependently predict or mutually predict both their own and each other's future liking sentiments. And that helps us understand how these idiosyncratic sentiments become dyadically coupled. I could spend 20 minutes just talking about limitations of this study, so I'll be brief and allow you to do that in the questions. But there is obviously a very specific social context of the participants in this study that is very different from lots of other kinds of groups. We are doing some follow-up studies to tease that apart. And as I mentioned before, the causal mechanisms here are not clear. We're not manipulating these kinds of brain responses between people, and so we don't have the ability to say that they are causally influencing liking at the end. I just want to point out one kind of neat future direction that we're going with this study that the PSSN program has allowed me to embark on, which I think is just awesome. This is a collaboration with a research team in South Korea. I'll be going there in April to study a group of elderly farmers. They've done longitudinal network studies of about 500 of these participants who have known each other on average about 40 years. And we got grant money to scan about 80 of them and have a much larger sample, a totally different kind of social context, something completely different from what we've been looking at now. Obviously, this kind of work, it would be ironic if I didn't recognize how important the outcomes of this work are, how dependent they are on the social inputs here. So I'd really like to thank Daphne and Peter for their, you were lucky that you got to see this and not the practice talk. The collaborators on this particular study that I talked about today from neuroscience, psychology, and sociology, the participants of this study who were amazing on their one day in New York that they agreed to be scanned at midnight for this crazy study, and all of the PSSN scholars and the committee and everybody for making this kind of work possible. This is not the kind of work that gets you a job a year out of grad school, which is the way that a lot of our decisions have to be made as young researchers, and I'm really grateful, like the other scholars are, for the opportunity to get to do weird kind of out there work that's multidisciplinary, that takes a long time to do because it's really fun. So thank you all for your attention.